will be more interesting to record the solution, but of course I will record the solution later. Okay. So what's going to be this practice about the differential gene expression? So we are going to do 2 exercises. So we will be working more or less half of the morning with this, because I would like you to test 2 different methods. One of them is the common one that you receive from ComRBAN. So you don't want to complicate your life. So that is a very convenient method. Or the other one is this SEC2, that is, as I said, one of the better in terms of recognition. Okay, so I'm going to tell you, so the exercise will consist in a lot of the data filtering. We will talk a little bit or a lot about this quality control, and then the differential expression test. Because all these things are something that you are going to run in R. I already specify what are the commands that you can run, okay? So you only need to open R and just continue with these things. So I think that at least almost all of you have some knowledge about R, so that should be doable. So remember that the paper that we are going to work with is this chromatin remodeler DMD promote hybrid vigor regulating salicylic acid metabolism. So at some point they did RNA-seq analysis with a mutant and also with these samples, that is Columbia samples, C24, that is a different accession, and then do different type of hybrids, Columbia C24, and just then the opposite, Columbia C24 Columbia. And of course, we want to test if we have differential expression between the hybrids and the 2 parents. So when we are thinking in comparison, we can compare them. The summary of what is the thing that they did is here, no? So they did the sequencing. So they are working with seedlings, okay? And then did the trimming and the alignment with Top Hat 2. We didn't use Top Hat 2. We used a new version that was HiSat or Start. And then they use HT set count. That is one of the things that you were using. And then they use HR, okay? So basically, of course, we can have different comparisons. So we can compare Columbia with C24. We can compare Columbia C24 with C24 Columbia, again, with these ones, or as many as pairs as you want. No, indeed, we have 1, 2, 3, 4, 5, and 6 comparisons if we want. So that will be one object. But the object that we wanted to use is to see if hybrids have differential expression compared with the parents. So for example, if these 2 parents has a very different expression, maybe it's not worth it to compare them. But if the 2 pairs had more or less the same expression for some specific genes, it's worth it to see if you have them, because you have these hybrids, you have a much different expression, that you have a transgression in the expression. It's expected that when you have 2 different expressions here, you should have here the average, but it's not always the case. So we are going to test for that. So we are interested to compare only these 2 groups. If we put together Columbia C24, so it means that you may have a lot of variation. We are going to compare with this to see if you have a differential expression. And as I mentioned before, the first pipeline that we have been using is this HiSat2, SlimTie, and Valgo. You already run SlimTie, or maybe you didn't finish, but I hope that you finished during the weekend. So you will have this transcript level expression. Okay, so you will have the results. So at some point you should be here in these SlimTie read tables. So the thing that you're going to do basically is copy the read tables in your computers and then run Valgo to check the differential expression. So if you are more interested, this is a good manual, a good tutorial here in this link. So you always can go there. And of course, before you do that, you need to set up your system. So the first thing that you need to do is transfer the SlimTie output from the server to your computer using R, okay? If you don't have the SlimTie output, just let me know. and I will copy and paste in the chat the location of mine, so you can copy everything in yours, and then you can transfer everything with FileZilla, okay? So just let me know. How many of you will need to copy these files? Okay. As soon as I have more than one, perfect. So I will copy them. So so as soon as I finish presenting the exercise, I will give you the link, and then you can copy from my directory. One question: do you prefer for me to copy from the directory, or do you prefer me to zip the file, put it here on Teams, and you don't need to use FileZilla? What do you prefer? Do you want to use FileZilla? Either is fine. Okay. Zip. Okay. So I will zip them. I would prefer to use FileZilla. Okay, so I will give you both options. Perfect. So that is the first step. Just copy the files. The second, of course, is open RStudio and upload the library Valgo and R color viewer, gene filter, and dplyr. Okay, so you need to have 1, 2, 3, 4 packages. And also, my recommendation is set up the working directory in the same one that contains the directory with the string type results. So my recommendation is usually you can navigate through this part of the window, so through files, you can navigate until you reach the directory that contains the files. And then in more, or at least in Mac is in more, probably you will have something similar in Windows, click this set as working directory. So that will set as a working directory, for example, the file where you have a string type. Okay, so that is an important part. The last thing that you need to do before you continue is you will need to set up a tabular text file that is called Data. I can copy that if you want to, in which you are going to tell the program how the samples are organized, what is the experimental design. So in this case, we will have several things. So we'll have an ID, and the ID should be the same than the name of the files or the folders that contains the expression data. So we will have this. Then accession. I wanted to have accession also as a variable. So we have C24, C24 Columbia, Columbia C for C24, and then Columbia. And then we will have the type. I put them pure lines or hybrids, and we will compare pure lines against hybrids. And of course, we have the replicates. No? So this is a tabular format file with 1, 2, 3, 4 columns. That is essential. Okay, so that is the preparation that you need for the exercise. Okay, and as I said, I'm going to tell you the end of the movie. Okay, I'm really sorry. If you want to move through the movie, I'm going to tell you how it's going to end. Because for me, the idea here is that you can follow the process, but because this data set has some difficulties, I will tell you about that now. I just prefer you to just follow all the different steps, and we can discuss why. Okay, because if I'm not going to tell you anything, at some point you are going to hit the rock thinking that there is an error. Okay, so the first is data upload, and as I mentioned, this is the commands that I'm going to give you. So if you are in the right directory and you have the phenodata txt file, so the first thing that you need to do is upload the experimental design, as you can see here. So I'm assigning to this R variable, I'm reading the file with read the limb. And then also I am sorting them. Okay, this is important because if you don't sort them, you will have problems when you are trying to create the Valgo program object. So that is the prep. Then you have, then is when you really upload the expression. No, you upload the expression that comes from string type using the Valgo. So you use the R command Valgo. So you say, okay, so my directory in my case was a directory called Valgo, but of course you can put any name. sample pattern. All the samples start with alpha, so that's the sample pattern. And then the P data is the pheno data that you can see that is the object that I created here. So that will create the bulk object. Once you have the bulk object, you can get some stats with just typing VG, that is the name of the object, or sometimes with summary. If you want to get the gene expression, is GS, or you want to get the transcript expression, is TX. Okay, so basically you can get the some stats here. Don't worry, because when we resolve the exercises, I will go to this step by step. Once you have the data uploaded, the next steps will be to filter the data. Sometimes it's not convenient to put all the data with that. So for the data filtering, it's simple. No? The thing that we want to do is we want to keep only the transcript with expression of the sum, all of them, of 1 FPKM or more. So to do that, we are going to use the function subset. And basically, the expression is of background of the bulk object. We use this row bar text expression of more than 1. Gene non-subset too, because we want to have a subset. Okay, so that will be kind of the filtering. We are not going to do that, but remember that I told you that if you want, you can split things, and then you can compare things splitted. So if you want to say, I want to compare just some subset. So imagine that you want to compare only Columbia with C24. You only want to compare the pure lines. So the thing that you can do is you can create a subset with only these two pure lines, and then compare C24 against Columbia. So that will be a pairwise comparison. So if you want to do that, you can use this function. We are not going to see that in the exercise, but just I put this for your records. And then once you have the filter data, of course, you can test again the summary and see what is there. Okay, now becomes the quality control. And as I said, I was doing this during the weekend, because when originally I was selecting the data sets, I wanted to select a data set with 3 replicates, where the 3 replicates have more or less the same number of reads. That was fine. And then with different accessions that we can do these pairwise comparisons in kind of in a different way. No? So I thought, well, this experiment was good. So you will see what is pop up from this. So the goal, as I said, is to visualize different parameters. So the first thing that you do is to visualize the expression distribution to see if you have more or less the same distribution. Remember that to test differential expression, as I mentioned in the presentation, you need to have similar distributions. If you don't have similar distribution, you cannot test expression. So the first is to check that we have similar distribution. The distribution may not be the same genes. Okay, that is the thing that we want to see. We want to see differential expression between them. But the distribution, okay, how much we have of everything, it should be more or less the same. The profiles should be the same, even if the identity of the genes are not. So to do that, first, we need to get the gene expression from the bulk objects, so as a data frame. So you use this gene expression function. Then you want to change the column names. So for example, you want to remove this FPKM, that's because it makes the names much longer. So you can remove it with that. You want to define a palette of colors. So I define this one for our case. We have 3 reds, 3 oranges, 3 salmons, and 3 greens. That will be, if I don't remember, it will be C24, C24 Columbia, Columbia 24, C24, and then Columbia. We will also change the names to have some short names to fit better. So that is the thing that we do to do that. These 2 lines are the command. And then finally, we want to do a box plot with the distribution. So this is how should look the box plot. As I said, I'm telling you the whole movie. I'm really sorry. And as you can see here, the first quality check for the samples looks fine. Okay, everything has more or less the same distributions, as you can see the average, and with more or less the same percentage of outgroups. So it looks fine. Of course, we don't have low expression values because we filtered them, but as I said, so that is a good signal. Then the next thing that we want to do, of course, is compare between replicates, because replicates should have more or less the same expression values, okay? So you want to calculate the correlation between them. So for example, in this example that I put here to you, we select replicate 1 of C24, we select replicate 2 of C24, and then we plot. And then we create an upline that will be a diagonal to check if everything adjusts to there. And then we calculate the correlation between the expression, and we put the correlation in one corner. So, and that is when this is starting to have problems. As I said, originally I read the paper, I thought that was a good tool, and then I was expecting that would be good. But my surprise was that when I ran this correlation value between the two replicates, the R, the correlation value, the R squared, was 0.18. Okay, it should be 0.8 or more. 0.7 is bad, but this is terrible. Okay, this is terrible. So, and one thing that I like from the exercise is that still you will see, because sometimes when we do these courses, we see everything in perfect conditions. So this is a good example of what happened where they are not, okay? So we are going to follow this line of problems. So you see this low correlation value, and the question is why? So we just compare two samples. So the next thing that you're going to do is you're going to create a matrix compared with all the samples to see if this is something that happened only between these two replicates or is happening everywhere. So to do that, this is the code that you are going to use. You will create a correlation matrix empty data frame using this command. You will put some names, and then you will use this loop to fill the matrix. And once you have that, you are going to create you are going to visualize them with a heat map. And this is how it looked like. Okay. And the first thing that I can tell you here is that all the replicate 1 samples clustered together, and then you have a mix between replicate 1, replicate 3, and replicate 2. But the thing that clustered together doesn't make sense. So here it's clustering together Columbia for C24, Columbia or C24. And then you have another cluster that is Columbia 24 replicate 2 and Columbia. And then here you have a cluster of 3 that is replicate 2, replicate 2, replicate 3 of C24 Columbia, C24, C24 or Columbia. So as you can see, this doesn't make sense at all. Of course, when I did that, I thought, well, maybe, sorry, I just, so I was preparing the exercises, I used the wrong name. So it will be as simple as change the names and we can move forward. But when I went to the MCBI, everything looks fine. Okay, so when I went to the MCBI, I checked the different runs with all the different samples. And again, this is kind of, one moment. Sorry, yeah, I needed to check this email because someone is telling me that the server that we are using okay. Yeah, they're telling me that the server has temperature issues. So, well, it's good that we are not going to work with the server today. As I said, we will work with the data, so it's fine. But as I said, I needed to check that. Okay, just let me go back to this problem. No, so I went to the NCBI. We see that, yeah, the samples had the right name. So it looks like the authors that upload the samples in the NCBI, they upload them with the wrong labels, hypothetically. So the thing that, of course, when you need to do that is, sorry. So it looks like that was the problem. Okay. I think I have another slide explaining what's the problem. Okay. So that is what it looks like for this correlation value. The third thing that you can do from the correlation value is, okay, so let's not look for correlation, but then look for distances, okay? And when you look for distances, one common plot is this Mds Distance Plot, okay? So when you want to do this MD distance plots, the thing that you do is you first transform the correlation matrix in distances, you scale them, and then you can plot just following these commands. And this is how it looks like, the distances. And again, we have the same problem. All the R1 samples are in one corner, and then we have some R3, R2. So this doesn't look good either. Samples should not cluster by replicates, but should cluster by samples. So I went back to the problem. So I went to the original paper to see if they mislabel something. And into the table that I have, you can see that, yeah, looking into the data that we have been pulling out, we have the right names, you know? So Columbia replicate 1 has 52 millions of pin reads. We have that. By the way, they don't upload the raw reads. Columbia replicate 2 and replicate 3, 44, 42, 44, 42. So yeah, we have the right names. So they did a mistake somewhere. By the way, they published the paper. That is the sad story. Okay, so that is the important lesson about the quality control. That doesn't mean that we cannot continue with the exercise, and we will. Okay, but an important note here, okay, of attention is you will see that we still can do the whole complete absolute analysis of this being wrong. Okay, so we will obtain differential express gene. We can obtain Go terms. We can obtain clusters, and still the experiment is wrong. Okay, so it's why. So the important take-home message here is that this quality control is essential. If for some kind of reason you switch a sample, okay, if you don't do this quality control, you will not see it. And another important thing is you can see a lot of publications, okay, and we usually, as a referees, we don't ask for this kind of qualitative control, but we should. So all the papers, all the publications that we have about RNA-seq should have in the supplementary data these graphs. Because if you don't have in the supplementary data these graphs, you don't know if they are doing the right thing, okay? So as I said, that is the note of attention. Okay, so that comes the differential expression test. That is the most important part. And you will see that this is not a big problem. So basically you run a test. So you have you run this test tool on your object over the gene. You cover it by type, and you want to get the false change. And you also want to have these FPKMs over the FPKMs. So you do that, and then you have your test. Of course, you can check how many genes do you have, just checking into the column of p-values, checking how many of them are greater than or smaller than 0.05. And you will see that you will have some. And then you can get this table if you want to put the final results in a table. No? You can output this into a table later. So to retrieve the significant genes, for example, as I said, you can get the significant asking how many of them have the p-value of 0.05 and then counting how many of them do you have. And you will see that you will have. So even if the design is wrong, you will see that you will have. And then you can plot the results. Okay, so that is another example, and you should have something like this. So even if you don't have the right design, you will have a differential express genes. Then if you want to have a final table that you want to export to Excel was in CSV. So this is the command. So you, of course, are going to get the gene expression table from gene expression. Then you need to change the row names to the IDs. And then, for example, it's convenient, no, because this table doesn't give you averages. So you can calculate the mean and the standard deviation for each of the replicates, as you can see here. Then you can match the table with the differential spread genes, and then you can write everything into a CSV outside R, just for your records. So that will be for the differential expression test. Okay, questions about the first part of the exercise? I can see a lot of rising hands, but I'm not sure if it's that, yeah. Oh, okay, I cannot see them. Questions that you have about the exercise?
I have a question.
Yeah.
I have a question. In the finodata.txt, we have 4 columns, and we always have these 4 columns, or I mean, accession and type are mandatory, or
well, it's not mandatory, but you will see that we are using them. So, for example, so of course you can create whatever you want. So mandatory is IDs, and IDs should be the same ones. So well, okay, mandatory is IDs and replicates, and they should be just the same one that's your experimental design. Then you can use accession, type, treatment, whatever name do you want. But remember, for example, in our case, we are using here type, and then when we are running the differential expression test, we use here in covariate, we put type. So if we have instead, we want to test if the covariance instead type is treatment, we should put there treatment. Or if we want to test accessions, we will put there accessions. So you can put whatever name do you want in the pheno table, but then you will need to use the same name to do the testing.
Okay, yeah, thanks, clear.
Okay. Any other question about the disappointment of finding something that is not right? You know, originally I thought, well, this is a disaster, but then I thought, well, maybe it's not, because really this is a really good exercise to go through the whole process and show you that still you can get differential express genes even if it's wrong. Okay, and that is a very powerful lesson here. Okay, so that is a powerful lesson because that means that, as I said before, qualitative controls are essential. If you don't do it, you don't know you have something right or wrong. It's like doing a PCR and doesn't have a negative control. You get the band, but you don't know if the band is the thing that you should have or not because you don't have a negative control. So that is the same thing. Qualitative controls for RNA-seq analysis is essential because it can put the results in perspective. Okay, so that is for CAM-RBAM. The second exercise is with DISSECT2. So, and again, remember that the process with this is star, HTSEC count, and DISSECT2. Of course, one option is that we have all this wrong data because CAM-RBAM is not giving us the right results, or maybe it was a problem with links, or maybe it was a problem with the mapping. But you will see that not, because that is a complete independent pipeline and still give us the same results. So you process so remember, you will process the risk with star, and then we count with HTSEC count, and then we use DISSECT2 to have the differential gene expression tables. So you want to have access to a good tutorial. I think that this is a good tutorial about DISSECT. So you can go to this link. Okay. Again, the first thing that you need to do is you need to copy the HTSEC count output in the server using FileZilla, or I will share with you where you can find them, or I will upload them here. The next thing that you need to do is you need to prepare a sample table object. Instead, use an external object, okay? That is this pheno table. You can create your own table in R. And for some kind of reason, that is the thing that they suggest here. So to do that is, of course, once you set up your working directory, remember that is essential. You can get a list of your sample files using this command. So you say artha with grep, you say artha just to select the name of the files, list files in the directory that you are. You should have only that, and then say valid true, so that will keep everything in a vector. Then you have sample conditions. So sample conditions will be, remember the right colors. We have pure line, pure line, pure line, hybrid, 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 and then pure line, pure line, pure line. Okay, so that will be sample conditions, second vector. The third vector that we want to have is sample names. So we will get to these sample files names, and we will remove this prefix of htsecCount.count, and also we will remove the artha. So that will give us the sample name. And then we build a sample table as a data frame with all these things. We have sample name, file name, and the conditions that are the three vectors that we create there. Then we need to have a factor of and the condition, because that is not a simple vector, need to be a factor. So to do that, you call factor and you rewrite condition with that. So that is to create the table. Then to upload the data is simple. You use DDS, well, you create the object just with the function dissectData set from htsecCount. You have sample table, the sample table that you create. You are calling the directory. Sample table is going to tell what are the file names. And then for the design, we are testing condition. Okay, we put condition, we could put type, so name, the same thing. And then we can get some stats if you want. The second will be the data filtering again. So in this case, because we have counts and not FPKMs, we are going to select transcript with the sum of counts of 10 or more. So this is the command that we are going to use, and then we are going to keep them. And again, we can get the summary. And that will be for data filtering. One difference with this is that you do the differential expression test first, and then you do the quality control of everything at the end. At the end is the same. I mean, it doesn't give you much change. Do the differential expression and do the quality control of do the quality control first to be prepared about what is the thing that you want to find. The differential expression is very simple. You run first dissect on the object, and then you that results. Okay, because we already specified that we were comparing these conditions, then just results, and then you check the result with how many of them have the p-value. Keep in mind that we are going to use p-value just to obtain differential expression. Of course, if we use the p-adjusted value of the FDR, we will get 0. Okay, so that is expected. And then the quality control. So quality control that is suggested with the dissect package are different. So first, you have this map plot, and where you compare the fold change, and then you have the normalization counts. In blue should appear the differential express genes, at least that's with the p-adjusted value, but we don't see any, as you can imagine. The other thing that they suggest is going, for example, to the gene that contains the lower p-value, okay, and then test. And as you can see, it's interesting that as you can see, we really have some differential expression here between the hybrid and the pure line. Okay, so they kind of overlap, but mostly, I mean, you can see the difference. Okay, the problem is that, as I said, the sample names may not be right. And then the final part, of course, is to do this heat map. So this is the commands that you need to run to have the heat map, and this is the result of the heat map, and it's just the same of the results that we get with CAM-RBAM. Okay, so you can see the replicates one clustered together and then the rest clustered together. Okay. Okay, so that is the exercise. Questions before I leave you with the exercise?
Yeah, I have a question again.
Yeah.
So we said that the SEC2 can analyze just pairwise samples, but we can clusterize more samples of the same condition to analyze two different conditions. It is clear. Okay. Where is the passage where we clusterize these samples? Because they are those were a lot of command, and I'm not very familiar with R.
Sorry, can you repeat the last question?
Yeah. What is the command to clusterize more samples?
So, well, in this case, we don't have a command to clusterize. So the thing is, again, the key here is the table. So in this table, in sample condition, is when we are cluster them. So in this table is when we are talking, we don't divide them by C24 or Columbia or the hybrids. We are dividing them in two conditions. We have the condition pure line and we have the condition hybrid. So here,
oh, nice, yeah.
Here is when we cluster this sample. We don't have a command. It's the design who tell DSEC how should cluster the samples.
Okay, nice. Yes. Thanks.
Yeah, and then here, when you are uploading the data, you are telling DSEC that is condition who's going to have the design of the comparison. Okay, any other question? Okay, so you don't have any other question. I'm going to, well, first I'm going to stop the recording. Let me see.